Ever wondered how chromosomal deletions impact genetic diseases? In the realm of genetics, deletion refers to an intriguing chromosomal anomaly that results in the loss of genetic material. This loss can give rise to a wide array of genetic conditions and diseases. It's a bit like removing a chapter from a book. Suddenly, the whole story can change. Over the years, advancements in genomic technologies have allowed us to not only identify these deletions with precision, but also to understand their character and behavior. This has significantly broadened our understanding of how these deletions impact our genetic makeup and their role in disease development. It's like having a magnifying glass that allows us to closely examine and comprehend the molecular mechanisms and the functional impacts of these deletions. As we continue to explore this fascinating world of genetics, let's delve deeper into the mechanisms that lead to these deletions. A myriad of mechanisms contribute to deletion formation. This vibrant tapestry woven by our genetic code is not infallible, and sometimes a thread goes missing. A deletion occurs. One of the primary culprits behind deletions are DNA double-strand breaks, or DSBs. These are like fractures in the genetic material, occurring spontaneously during the process of DNA replication or induced by external factors such as radiation or chemicals. Now, our bodies have complex machinery for repairing these breaks. Two key players in this process are non-homologous end joining, or NHEJ, and homologous recombination, or HR. Picture NHEJ as a quick fix. It's like taping the broken ends together, albeit sometimes imperfectly. On the other hand, HR is more like a skilled craftsman, using a sister chromatid as a blueprint to accurately mend the break. However, when these repair systems falter, the result can be a loss of genetic material, leading to the formation of deletions. Imagine it as a sentence in a book where suddenly a chunk of words go missing, altering the meaning of the sentence. That's exactly what happens to our genetic code. Moreover, our environment and lifestyle play a significant role in this process. Exposure to certain environmental factors and mutagens can cause DNA damage, thereby increasing the likelihood of deletions. It's akin to living in a city with a high crime rate, where the chances of being robbed are higher. The interplay between these biological and environmental factors sets the stage for deletion formation. It's a dance, a delicate balance of creation and destruction, repair and damage. And when this balance is disrupted, deletions occur, leaving an indelible mark on our genetic code. And so, the mechanisms behind deletions are multifaceted and complex, involving both our genetic machinery and the world around us. It's a testament to the intricate and dynamic nature of our genetic code. Yet, these mechanisms in turn have profound effects on chromosomal function, altering the symphony of life in ways both subtle and profound. Deletions can significantly alter chromosomal function and gene expression. When essential genetic material is lost to deletions, the repercussions are far-reaching. The normal activities of a cell, the smallest unit of life, can be disrupted. This disruption can, in turn, lead to an array of developmental anomalies. It's like removing an important cog from a well-oiled machine. The entire system grapples with the sudden change, trying to adapt and compensate, but the result is often a breakdown. This breakdown is not just confined to a single cell. It can manifest in the form of cognitive impairments, affecting an individual's ability to think, learn, and remember. Deletions can also create a susceptibility to various diseases. Imagine a fortress with a compromised wall. That's what a cell with a deletion is like, vulnerable and open to invasions by disease-causing agents, including the dreaded cancer. Now, let's delve a bit into the concept of haploinsufficiency. It's a term that sounds complex, but it essentially means that a single functional copy of a gene is not enough to maintain normal function. When deletions occur, they can lead to haploinsufficiency. Picture a pair of workers lifting a heavy load. If one worker disappears, the remaining one struggles to lift the load on his own. Similarly, when one copy of a gene is deleted, the remaining copy may not be sufficient to carry out the gene's function, leading to various phenotypic manifestations. These manifestations are often associated with the disorder related to that particular gene. In essence, deletions are like missing pieces in the intricate puzzle of life. Their absence disrupts the beautiful picture that our genetic material paints, leading to a cascade of effects that impact our health and development. But there's a silver lining to this cloud. The better we understand these impacts, the more effectively we can devise strategies to combat them. Understanding these impacts opens up therapeutic avenues. It's a testament to the power of knowledge and our relentless pursuit of understanding the world within us. The knowledge of deletion formation mechanisms holds great therapeutic promise. This statement holds the key to our hopes for a future where genetic disorders caused by deletions are no longer insurmountable challenges. Let's consider some of the potential interventions on the horizon. One of these is gene therapy, a medical field that has already made a significant impact in treating a variety of diseases. This approach involves introducing, removing, or altering genetic material within a patient's cells to mitigate or cure disease. For disorders caused by deletions, gene therapy could potentially replace the missing genetic material, restoring normal function. Another promising frontier is gene editing technologies, like the revolutionary CRISPR-Cas9 system. This tool allows scientists to edit parts of the genome by adding, removing, or altering sections of the DNA sequence. Think of it as a molecular pair of scissors that can snip out the problematic parts and replace them with healthy ones. In the context of deletions, this could mean precisely targeting and correcting the deleted segment, potentially reversing the effects of the chromosomal abnormality. But it's not just about correcting deletions after they've occurred. There are also strategies aimed at preventing or repairing DNA damage in the first place. For instance, DNA repair inhibitors can be used to enhance the cell's natural defense mechanisms against DNA damage, thereby reducing the likelihood of deletions. Similarly, radioprotective agents can shield cells from the harmful effects of radiation, one of the environmental factors that can induce deletions. These are just a few examples of how our growing understanding of deletions can be harnessed for therapeutic purposes. But as with all scientific endeavors, there are still many unanswered questions and challenges to overcome. Continued research into deletions and their role in disease pathogenesis is set to revolutionize the treatment of genetic disorders. Deletions, once an elusive and misunderstood aspect of our genome, are now at the forefront of genetic research, promising to unlock new ways to prevent, manage, and treat a wide range of diseases. The future of genetics, it seems, is not just about understanding our DNA, but also about learning to rewrite it.